Okay, thank you very much, everybody. I'm going to talk about just broad strokes. How do we personalize medicine in cancer overall? And then actually, my colleague, Dr. Shaji Kumar, is going to take it to the next step and show you how he is leading some of the leading efforts in trying to personalize it to individuals in myeloma. So personalized medicine. So as we just heard this morning, and this slide was already shown by my colleague this morning, that you do right now treat myeloma fairly homogeneously. The fact is that yes, at the beginning you do try to figure out if a patient is transplant eligible or not. And if they are transplant eligible, as we went through this morning, you go through a phase of induction, transplant, maintenance therapy. And then for those who are not transplant eligible, generally we give these days mostly an attempt to give continuous therapy with multiple agents. And unfortunately, yes, in most cases, patients do still relapse. And then we go to fairly standard options or trial options, as we were discussing this morning, for relapse disease. But it's the same options, whether you're sequencing CAR T cells, it's for all patients. And that has a good advantage. The thing is that you are able to give patients therapies irrespective of their individual genetic makeup. But again, can we be precise to each given individual? And that's what we're going to talk about briefly today. So the fact is that as far as trying to personalize therapy, one of the ways that has been out there for a long time is to check the sensitivity of the cancer cells to certain chemotherapies. This is like the analogy: if you have a bacterial infection, the microbiology lab will tell you what antibiotic your bug is sensitive to. So attempts in this area have been ongoing now for well over 20 years. And yes, we're getting better at it, but I think we still don't, have a way to scale this model up, wherein we can identify for a given individual, if we take their, if we take their cells, can we test in the laboratory what drug would be best for a given patient? And in the past, we have had models where we just take the cells and we put certain chemotherapy drugs into different wells, pipette it into a petri dish and see which cells die. And that model doesn't translate into actual effectiveness in humans, because the cancer cell in a human being doesn't live in isolation. It lives in an environment of immune cells, other structural elements in the bone marrow. And so now there are models that are emerging. Some of the work is actually from a colleague of mine over here, where we are able to not only take the actual cancer cell, but also the environmental cells of the individual and grow those out in what we call 3D models, and then use that to predict for drug sensitivity. And the fact is that at least in the petri dish, you can distinguish what patients are likely to respond to, but is that going to work actually in the patient? That we are not yet clear about. And to do these kind of trials, it is very difficult because these things do take several days to grow out, and by the time you get the reports, sometimes it's been several days to a week or two, and often patients need treatment right away. So these things, yes, there is still ongoing interest in. These are technologies that will continue to be refined, and but what we do today is personalized treatment based on individual patient characteristics, perhaps not their disease characteristics, based on their age, their comorbidities, what their psychosocial status is, how healthy they are, what they've had in the past, patient preferences. So we are doing some kind of personalization, but this is not what we really think of when we talk about personalized medicine. And that is something that we are starting to unravel a little bit at a more macroscopic level. So these guidelines from the Mayo Clinic, where Dr. Shaji Kumar works, who will speak after me, have been put out and are regularly updated, wherein you take information from the patient's FISH test and cytogenetic test that nearly all patients with myeloma have done, and then give patients specific treatments based on what their cytogenetic profile is. Now, this is not drawn from any concrete prospective trial data. It is an interpretation of all the data out there, and it is something that is sort of a consensus statement of the physicians at Mayo Clinic. They have a lot of physicians at the multiple Mayo Clinics to try to have a homogeneous approach among their faculty members. And a lot of people do look up this, even in the community. But I must say that with due deference to Dr. Shaji Kumar, that not everybody totally agrees with these guidelines, but they are out there. They tell you that these are ways to start to unravel the problem. And the first test that is probably going to have some utility in our FISH panel to come to clinical utility is what we call 11-14 translocation, which is a translocation that happens in about 15% of patients with myeloma, and therein this drug called venetoclax has been shown to have a much higher cancer response and a duration of response. And this again is work that Dr. Kumar has done and led at an international level. In one trial that he heralded, there was venetoclax given to all patients. That didn't lead to good outcomes for all patients, but when you look at the specific 11-14 translocation subset, this is something that patients respond to well. So if you have an 11-14 translocation, even today, though venetoclax is not FDA approved, either on a trial or through a compassionate use program, we are able to usually treat patients with venetoclax and have very good results. I've had patients who had every known therapy, who've actually had even CAR T cells and then relapsed and gone on to getting venetoclax and have had disease control even post CAR T cells if they have this 11-14 translocation for 1.5 to 2 years. So this kind of information is going to have clinical utility. The other way that people want to see if they can personalize therapy, which some individuals are starting to do, is based on the MRD, or the measurable residual disease analysis. This is something that several of you probably have heard of, that we can nowadays test your bone marrow and find one cell abnormal in 1 million. And the fact is that we know that patients who are MRD negative do better than those who are MRD positive. But what we lack from prospective scientific data is how to use this information for decision making. So if you are MRD positive and another person is MRD negative, after say 4 months of treatment, the person who's MRD positive is going to not do as well as the MRD negative person. But what we don't know is if we change the therapy of that MRD positive person to make him MRD negative, will that patient then catch up and still have the good outcomes? Or if you are on maintenance therapy, which is very common now in myeloma, and you often have maintenance therapy going for years and sometimes are having even side effects, can you, in say people who are sustained MRD negative, that is are MRD negative for a fixed period of time of 1 year or 2, stop the treatment and still have the same results in those who are MRD negative as continuing treatment and going on treatment for years? So those are questions that are being addressed in prospective trials. And so MRD to personalized therapy is something that some institutions and individuals are starting to practice, but that is based on what they think is their best hunch, their best guess of how the disease should be treated. It is not yet based on sound scientific evidence, but it's already happening in certain institutions. They say if you're MRD negative, then maybe you don't need a transplant at the beginning. Maybe you should wait till your disease relapses. Those are, again, things that from proper scientific trials will hopefully learn in the next 5+ year time frame. But then truly personalization is trying to look at the genetics of the cancer at a much more finer level, at the level of genes. So FISH testing and chromosomal testing is something that you look at under the microscope, 200 cells in a FISH test, and you are probing for certain genetic changes that you know are prognostic. And that, as I said, is clinically applicable. But genomic testing, where you're actually doing the screening of all the genes in an individual's genome, is a finer level of granularity. And in the case of a lot of different cancers, this information is already actionable. For example, in lung cancer, in certain leukemias, if you have certain mutations, you are able to tailor therapy, and you know that patients who get tailored therapy do better than those who get just random pick. Okay, you've not had a CAR T, let's give you a CAR T. If you've not had a BiTE, let's give you a BiTE. So the issue in multiple myeloma is that we are at the moment not there yet. We have some kind of information that some mutations perhaps could be tackled with targeted treatments. But the issue is that in myeloma, I'll just go back one second. So in myeloma, first of all, it's a somewhat rare disease. There are only probably 120 to 150,000 individuals at any given time, in contrast to millions of people with breast cancer or lung cancer out there. So getting the information is, you got small data sets. But what we have now with hundreds of patients sequenced, known that there are certain mutations that occur in myeloma, but they occur in relatively small subsets. In contrast to, say, leukemia, where a genetic mutation may be found in 30 to 40% of the leukemia, and then the drug companies can do trials to target that mutation. In myeloma, most mutations don't occur in any more than 5% of individuals. So if you have just a finite number of patients to do trials where half the people get a certain drug and the half don't, which is the gold standard of trying to prove that a new therapy is better, it's very difficult. It is not possible often to do these kind of trials. And the other thing is that, as was discussed earlier, these mutations may occur in what we call the founder clone, that the very first cell that became cancerous. In that case, going after that mutation makes sense, because then all the cells that arose from that original cancer cell will have that mutation. But in myeloma, these mutations that first of all occur only in 5% of cases often occur in what we call subclones, that they occur only in a part of those cells. And you can find out by current testing whether these are present in 5% or 50%. Certainly, if the more cells that carry the mutation, the more likely that you will respond to a drug. If you got it in 5%, if that is really the aggressive part of your cancer, maybe you'll have a transient response. But the fact is, for most patients, those are going to probably not be long-lived responses. So now we're going to even a greater level of granularity with current studies that are ongoing. So when we talked about this previous experience, we took part in this large study that was done, which was the initial sequencing of the myeloma genome with a number of other academic centers. What you're doing is you're just taking the whole cancer, all the cancer cells, and you're saying how many mutations are there. Now we have technology called single cell sequencing that is able to actually go down to a single cell level. So in this graphic here, each dot is a single cancer cell from the same individual. And so you can see that when you start going down to a single cell level, there is a great heterogeneity in the different cancers. So now you have resolution on what individual mutations occur in any given cell. And you can also know whether a single cell has 2 mutations, 3 mutations, or 4 mutations, whereas the older technology is able to just give you a bulk readout that some cell in this cancer carries this mutation. But at this level, you can say, okay, 20% of the cells have 5 mutations, 5% of the cells have a different set of mutations. So this is something that will probably lead to even greater ability to use this information for personalized medicine. And this just shows you some of the data that has emerged from this kind of technology, that when you look at the plasma cells from a given individual, you actually have very different populations of plasma cells, and these arise from, as we've been saying this morning, from B cells. And so the thing is that these are often the normal B cells, and there are now populations that have mutations that are specific to the late stage of the B cell, that tells you that perhaps those are the mutations that may be giving rise to the myeloma. And going after them probably makes more sense. And then the same technology now can be used to actually look at not only the cancer cell, but each and every cell in the microenvironment of that cell. So as we have heard elegantly this morning, that immune cells play a major role in regulating cancer. So now we are able to actually probe each and every cell in the microenvironment of that cell. This was technology that was not available even 5 years ago. And you're able to do single cell sequencing on every cell in the microenvironment and find out what are the mutations and what are the expression of different proteins in these various cells in the microenvironment. And what we are starting to see is that there are actually certain things that are emerging. This data that we have from our own institution shows that certain patients with a given mutation have a clustering of certain immune cells together, suggesting that if you have a certain mutation, not only is your cancer cell going to behave similarly, but actually the immune cells react to that in a similar manner. So these are obviously very basic scientific issues that need to be unraveled, and before you can use this information for therapy planning. Then the next level obviously is going to be what we call in silico engineering. So even if you get the information that you have mutation X, Y, and Z in a cancer cell, how do these different mutations interact? And that how the cell is affected by mutations often doesn't depend just on whether they have mutation in gene X or Y, but on what other mutations they have. So artificial algorithms, artificial intelligence algorithms are now being studied, and there are companies that are starting to build what we call these in silico avatars, where they, right now, when you do next generation sequencing and you get a report, the report you get is that you have mutations in gene X, Y, and Z. This takes it to the next level to say, if you have mutations in gene X, Y, and Z, this is what's going to happen to the cell. So these are in their infancy. There are only a few companies right now that are looking at this, but again, this is the wave of the future. And then again, there is this whole personalized medicine area that revolves around identifying what we call neoantigens. And this is something that, again, is right now being used in certain cancers, wherein you actually sequence the cancer of the individual and you figure out what mutations that the individual has. And actually, by using scientific algorithms, you can actually see how your immune system is going to respond to those mutations. So if you have a certain mutation, it's more likely that your immune cell can respond to that mutation versus not having that mutation. So this is actually the marriage between immunology and personalized medicine. So in certain disease states, pilot studies have already been done, like we did a study here in melanoma, the skin cancer, where people had, based on their genomics, identified that they have certain mutations, and scientists are able to tell us that if they have this mutation, they're more likely to respond to immune therapy. And so you can enrich the patient's immune cells to take out those cells that respond to that antigen, grow them, and then infuse them into the patient to act even more specifically against the cancer. So these are things that I think ultimately will come one day to myeloma. It's probably going to be 10, 15, 20 years away, but this is where I think personalized medicine may even meet immunology. The other thing to say, and this is going to lead into the next talk, is that because myeloma is a rare disease, and as I said, we don't have enough patients to answer these questions of personalized medicine, we're going to need to do what we call novel trial designs. Classically, right now, trial drug development goes through what we call phase 1, 2, 3 studies. Phase 1 is you have anywhere between 20 to 40 patients. You're trying to make sure the drug is safe. You get a little hint of whether the drug is active. Then if it is looking to be active, you enroll another 40 to 50 patients, and then actually at the full dose of the drug, treat those patients and see how active it is. And if it still looks promising, then you do what's called a full randomized study where half the people get the drug and half don't. And that's usually the evidence it takes for the FDA to approve a drug. And so that is often not possible when you're trying to do personalized medicine. As I said, in myeloma, a rare disease, doing such a trial could take years. And pharmaceutical companies are not going to invest in those kind of trials. So how can we go about it? And so there we come up with new trial designs, and this is something I'm sure Dr. Kumar is going to talk about. He's leading one of the largest umbrella trials in myeloma, where you actually get the different pharmaceutical companies, which is rare, to partner and say, okay, we're going to try to solve this problem together, and we will fund a single study where if you have a certain mutation, then you'll get a Pfizer drug. If you have a second mutation, you'll get a Moderna drug, or a third mutation, you'll get a GSK drug. And this requires great effort. I must tell you, Dr. Kumar is leading one of these studies, and it took several years, he can attest to it, I'm sure, to get these various companies to come together, their lawyers to sign off and say, we will fund this one study together. Because most studies are funded by one pharmaceutical company, and they want intellectual property, and this is all often a barrier to trial development. And then there's the basket trial design, where you say, okay, we've got a rare cancer, we got rare mutations. What we're going to do is we're going to just run a trial for a certain mutation. And no matter if you have lung cancer, breast cancer, or myeloma, if you have that mutation, we're going to run a single trial, and we're going to get a drug approved based on the mutation, irrespective of what kind of cancer you have. And now we have a couple of drugs that have, for the first time in the last year or two, been approved by the FDA using that strategy. So I'm going to finish here by saying that currently therapy for patients with myeloma is personalized based on patient characteristics. We are just starting to personalize therapy based on cytogenetics and fish characteristics. As technology advances, it is hoped that in the future, we'll be able to personalize therapy based on the molecular signature of the tumor and its microenvironment. Thank you very much.